Dear students, now we will talk about sensory organs. We will talk about organ of vision. So, organ of vision, according to classification, it's a primary type of sensory organs or neurosensory type of organs of sensory system. So, organ of vision, it includes eyeball and supportive apparatus of organ of vision. It includes eyelids with eyelashes, eyebrows, muscles which move eyeball, and also it includes lacrimal apparatus, which contains lacrimal gland and lacrimal ducts. And we will begin from eyeball. So eyeball has wall, which has 3 layers and also different inner structures. Wall of eyeball includes 3 layers. The outermost layer is fibrous layer. Here we can see it. It includes sclera and cornea. Middle layer is uvea. Here we can see uvea. It includes choroid. It includes ciliary body and iris. And the innermost layer is retina of eyeball. Here we can see retina of eyeball. It's receptor layer of eye which provides receiving of information about vision. And also we have inner structures. We have vitreous body. We have lens. And also there are chambers, anterior and posterior chamber. Anterior chamber of eyeball is located between cornea and iris. And between iris and lens we have posterior chamber. They are filled with fluid. And also we have optic nerve. So here we can see the outermost layer of eyeball. It's fibrous layer. It has transparent part, it's cornea, and non-transparent part, it's sclera. Next layer, middle layer, it's uvea, which has 3 parts. It's iris, ciliary body, and choroid. Here we can see them. And the innermost layer, it's retina, or nervous layer, or receptor layer of eyeball. It's retina, and it's continued by optic nerve. And also we have inner structures of eyeball. We have lens. Here we can see lens, and it's located near the ciliary body. There is ciliary ligament which maintains lens. And also we have vitreous body which fills the inner part of eyeball. And here we can see different chambers. It's anterior chamber which is located between cornea and iris. Iris has pupil opening. And between iris and lens we have posterior chamber of eyeball, and those chambers they are filled with fluid. So we have some transparent parts of eye. It's cornea, anterior, posterior chambers, lens, and vitreous body. And also we have wall of eyeball. Here you can see a histological slide which is called posterior wall of eye. Here we can see 3 basic layers, 3 layers of posterior wall of eye. It's fibrous layer. In this place it's sclera. Next it's uvea. In this place it's choroid. And the innermost layer is retina. 3 layers of posterior wall of eye. They are visible on this slide. And we will begin from fibrous layer, the outermost layer. It includes cornea and sclera. Cornea, it's transparent part, and it's continued by non-transparent sclera. So we'll begin from cornea. It's transparent part, the most anterior part of our eye, and it's transparent and it participates in the conduction of light inside the eye, and also it's very powerful lens of our eye. So here we can see the structure of cornea, and it has 5 layers. It has anterior epithelium, anterior membrane, substantia proper, posterior membrane or posterior limiting membrane, and posterior epithelium. Here we can see anterior epithelium. It's stratified, squamous, non-keratinized. Anterior membrane is located under this epithelium. Anterior limiting membrane or Bowman's membrane. Next, it's substantia proper. Next, posterior limiting membrane or descemet membrane, and posterior epithelium of cornea, 5 layers. Posterior epithelium is non-stratified or simple squamous, and anterior is squamous but stratified. Stratified squamous non-keratinized epithelium is anterior, and simple squamous is posterior epithelium. Next, anterior and posterior limiting membranes, Bowman's membrane and descemet membrane, and substantia proper. A special feature of cornea is absence of blood vessels. We cannot find here blood vessels, and it's very important for the functions of cornea because cornea is transparent and it's needed to provide transparency of cornea, and growth of blood vessels may cause different diseases of cornea, and blood vessels, they aren't needed here because cornea should be transparent. But how do cornea receive nutrients? It receives nutrients from teardrops, from this fluid which covers cornea, and also it receives nutrients from tears, and also it receives nutrients from vitreous fluid which fills anterior chamber. And here we can see cornea, and when blood vessels are growing inside the cornea, it causes diseases, and cornea isn't so transparent, so it's not normal condition when blood vessels are growing inside the cornea. So cornea should be transparent and avascular. And near the cornea we have sclera. Here we can see cornea and sclera. Sclera isn't transparent, it has blood vessels, and it has dense, irregular connective tissue. Sclera, it maintains the shape of eye, and it maintains inner content of eye, so it protects the eye. It's very important layer. It maintains the shape, round shape of eyeball. It has maintaining protective function, and it forms the eye outside. Sclera is covered with conjunctive. It's a mucous membrane of eye. It covers sclera, and inside sclera it has a lot of collagen fibers, which provide this density of sclera. And here we can see posterior wall of eye, and the outermost layer, its fibrous layer, it's called sclera. Next layer, antifibrous layer, next layer, it's uvea. Here we can see uvea, and it has 3 parts. It's choroid, it's iris, and its ciliary body. Here we have choroid or vascular layer of eye. Here we can see it. We have iris, which may have different shades, different colors, and also we have ciliary body, which has ciliary processes. It has ciliary muscle, and also ciliary ligament is attached to the ciliary body, and it maintains lens. Here we can see lens, and there are fibers of ciliary ligament, and those fibers, they maintain lens because they are attached to the ciliary body. And also ciliary body together with ciliary ligament and lens, they participate in the accommodation, adjustment of eye to different points of vision. So 3 parts of uvea or vascular part of eye. It's choroid or vascular layer, which is located between sclera and retina. Iris, which provides adjustment of eye to the brightness, and also ciliary body, which participates in the accommodation of lens, and also it produces vitreous fluid, which is produced by ciliary processes. So let's look at the structure of those parts. First, it's choroid. Here we can see choroid, and it's located between sclera and retina. It contains a lot of blood vessels which provide nutrition of retina of eye, and it's called a vascular layer. And also choroid contains a lot of pigment cells. That's why choroid has this appearance. So it's choroid, and it has numerous blood vessels, and they underlie retina and provide nutrition of retinal or nervous layer of eyeball. And here we can see bottom of eyeball. It's special method is ophthalmoscopy, and here we can see also blood vessels which are branched, which are visible here, and they also provide nutrition of retina. Next part of uvea is iris. Here we can see iris, and it has pupil opening which works like a diaphragm, and also iris has some layers. It has anterior epithelium and posterior epithelium, anterior intermediate layer and posterior intermediate layer, and middle or vascular layer. Vascular layer contains a lot of blood vessels which provide nutrition of iris, and also pattern of distribution of pigment cells in the connective tissue of anterior and posterior intermediate layers. They form special pattern and special color of iris, and all these features they result in the color and pattern of iris. It's very individual. And posterior pigment epithelium is continuation of pigment epithelium layer of retina, and later we'll talk about pigment cells of retina. Anterior epithelium isn't pigment as posterior, but anterior epithelium and epithelial cells, they may uptake granules of melanin, and they may contain melanin. So amount and distribution of melanocytes, of pigment cells and pigment inside them, it results in different color of eyes. And also distribution of blood vessels also participate in this. And between posterior intermediate layer and middle vascular layer, we have layer of muscles of iris. There are smooth muscles which form sphincter and dilator of pupil. They participate in the adjustment of iris to different brightness, and they provide dilatation, and they may make pupil smaller or bigger, and it acts like a diaphragm. So here we can see different color of eyes and different amount of melanin. It results in different color of eyes, and here we can see a case of albinism when pigment isn't produced in the pigment cells, and we can see pink iris because of presence of blood vessels. So it results in this shade of eyes. And in this case we can see heterochromia when eyes have different shade, different color. It's also abnormal condition. So here we can see a histological slide of iris, anterior posterior epithelium, middle vascular layer, anterior intermediate posterior intermediate layer. And here we can see sphincter and dilator of pupil. There are muscles of pupil, muscles of iris, and they regulate diameter of pupil, and it adjusts eye to different brightness of light. When light is very bright, sphincter works. When we are in the darkness, dilator works. And also those muscles are under control of parasympathetic and sympathetic nervous system, and nervous system regulates diameter of pupil of iris. So when light is bright, we can see contraction of sphincter of pupil of eye, and pupil becomes to be smaller, and it adjusts eye to bigger brightness of light, and a smaller amount of light enters in the eye, because too big amount of light may cause damage of retina, and we need this adjustment. And when we are in the darkness, pupil becomes to be bigger because dilator is working, so pupil is dilated. Next part of uvea is ciliary body. So uvea has iris, it has choroid, and we have also ciliary body. Ciliary body, it contains ciliary muscle. Also it has ciliary processes which produce fluid, aqueous fluid, which fills posterior, anterior chambers. This fluid is released in posterior chamber, and then this fluid goes through the pupil to anterior chamber. And here this fluid is removed. It goes to the Schlemm's canal, which is located between cornea and sclera. It goes here, and this canal provides removal of this fluid from anterior and posterior chambers. So ciliary body, first function of ciliary body is production of this fluid, and second function is participation in the accommodation of lens. Here we can see lens and its ciliary ligament or thin ligament, and it attaches ciliary body to lens. Ciliary body and ciliary muscle, it provides accommodation of lens. So here you can see lens. It's epithelial structure, and it contains modified epithelial cells, and it has nucleus and peripheral parts and capsule. So in the peripheral parts we have alive cells which are modified, but in the central part, in the nucleus, those cells, they are completely filled with special protein crystalline, and they lost their organelles, they lost their nuclei, so they are postcellular structures. And lens is very transparent, and also it has very big power of optic power, and it participates in the reflection of light and accommodation of eye to different points of vision. So it's second powerful lens in our eye. First one is cornea, and second is lens. So there are lens fibers, which are modified epithelial cells, and in the central part they even haven't organelles, they haven't nuclei, and they are completely transparent. And here we can see cells, peripheral cells are typical and they are alive, but central cells, they are postcellular structures and they have lost all organelles, or they have lost everything, but they are completely filled with transparent protein crystalline. And lens, together with thin ligament or ciliary ligament and ciliary bodies, they participate in the accommodation of eye. Ciliary ligament or zonular fibers, they are on one side, they are attached to the ciliary body, on another side they are attached to the lens. When ciliary muscle is relaxed, zonular fibers are under tension, and this results in the unaccommodated lens. It's flattened in shape. When zonular fibers are relaxed, lens is accommodated in relaxed condition. It becomes to be more thick. When fibers are under tension, it's flattened in shape. So when ciliary muscles are contracted, zonular fibers are relaxed, and lens becomes to be accommodated. It undergoes bigger optic power, and it is adjusted for the near target. Here we can see near target, and flattened lens is accommodated for the far target. When ciliary muscle is contracted, zonular fibers are relaxed, and lens is accommodated to the near target. When ciliary muscle is relaxed, zonular fibers are under tension, and it results in the flattening of the shape of lens, and lens is accommodated to the far target. So it's how accommodative apparatus of eye works. So here we can see accommodation to the near target and accommodation to the far target, flattening of the shape of lens. And also, next question is circulation of fluid inside the chambers. Here we can see ciliary body and ciliary processes. They produce fluid, and it goes to the posterior chambers. From posterior chamber through the pupil, it goes to the anterior chamber and goes to the Schlemm canal. It's aqueous fluid, and it provides circulation of fluid inside the chambers of eye. This fluid also participates in the conduction of light inside the eye. Here we can see this type of fluid circulation. This mechanism of fluid circulation. So ciliary body produces this fluid. It goes near the iris and goes through the pupil inside the iris and goes to the anterior chamber. There is a special canal between cornea and sclera which uptakes this fluid, and this fluid goes to the veins of eye. Next part, it's vitreous body. Here we can see vitreous body. It's almost avascular, and this structure of this vitreous body is a like structure of amorphous substance of loose connective tissue. There are a lot of different proteoglycans, a lot of water, so it has jelly-like condition, and it fills the inner space inside the eye, and it's transparent, and it participates in the conduction of light inside the eye. And next part of eye, it's retina. Here we can see retina. So we have discussed sclera, cornea, iris, ciliary body, choroid, lens, and vitreous body. And now let's talk about retina. Retina is a receptor apparatus of eye, and it works like matrix of cam. And let's look how is it working. So here we can see sclera, choroid, and the innermost layer is retina of eye. And the most important cells and the first cells which receive information inside the retina, there are photosensory cells. They are very specific, and they are located only in the retina of eye. So there are two kinds of photosensory cells. There are rods and cones. Here we can see rods and cones. So they have common features. They have perikaryon, so they are modified neurons. They have perikaryon, they have modified axon, and this part of cell is modified dendrite. This modified dendrite has outer segment and inner segment, and there is special cilia which connects them. So outer segment, inner segment, perikaryon with nucleus, and modified axon. It's structure of rod and cone. Those outer segments, they provide exception of information. They react to the light, and they have special membranous structures. In rods, we have discs. Here we can see discs, and they are completely enclosed. They are located inside the rod, and they have no connection with outer membrane. And in cones, we have hemidiscs. They are opened, and they are formed by outer membrane. Here we can see structural difference between rods and cones. Those discs or hemidiscs, they also are called lamella, and they contain optic pigments. In rods we have rhodopsin, and in cones we have iodopsin, special pigments which react to the light, and they play very important role in the light exception, reaction to the light, and irritation of rods and cones. So when light comes to those discs or hemidiscs, it causes configuration changes in rhodopsin and iodopsin, and finally it results in the excitation of rod or cone. So it's common features of rods and cones. But what are the differences? As you already know, the difference is a different type of optic pigment, rhodopsin and iodopsin, and discs and hemidiscs in rods and cones. But also there is functional difference. Rods, they provide black and white vision. They are very sensitive, and they provide vision in the darkness, in evening, or in the absence or in the lack of lighting. But cones, they need very bright lightness, but they are more specific and they provide colorful vision. They react to color types of light. So rods, they provide black and white vision, it's evening vision type of vision, and cones, they provide color vision. Here we can see rods and cones. They are first modified neurons or photosensory neurons which react to the light. But they aren't only cells in the retina, they conduct information to the next cells, which are bipolar cells. Here we can see bipolar cells. They have one dendrite and one axon. On one side, dendrite receives information from rod or from cone, and on another side, they have axons which send information to the following cells. So it's second neuron of retina of eye, bipolar cells. And look here, bipolar cells which receive information from cone, their ratio to cones is 1 to 1. One cone has one bipolar cell. But rods, they send information, some rods may send information to one bipolar cell. One bipolar cell receives information from some rods. So vision in darkness, black and white vision, isn't very specific because it isn't very precise because rods, information from rods is collected by one bipolar cell. But in cones, this vision is more precise because their ratio is 1 to 1. So first cells are rods and cones, photosensory cells. Second cells are bipolar cells. And also in retina we have third neurons. They are multipolar cells or ganglionic cells. They receive information from bipolar cells. So rods and cones receive information. They send this information to bipolar cells, and bipolar cells, they send this information via axons. They send this information to multipolar neurons. Here we can see multipolar neurons or ganglionic cells. And then they send this information to CNS. Their axons, they form optic nerve, and this optic nerve excites from eye and goes to brain. So how information in the human retina is conducted? When light comes here, it irritates rods or cones. They are activated, and wave of excitation goes through rods to bipolar cells and to ganglionic or multipolar cells. So first neuron, it's rod or cone, photosensory cell, next it's bipolar cell, and third is multipolar cell or ganglionic cell. So retinal neurons are photoreceptors, bipolar neurons, and multipolar or ganglionic neurons. But also we have two additional types of retinal neurons. There are horizontal cells and hemicrine cells. Why do we have them? Horizontal cells, they are located between layer of photoreceptors and bipolar neurons. Here we can see layer of photoreceptors and layer of bipolar neurons, and here we have horizontal cells. And between bipolar neurons and multipolar neurons, we have hemicrine cells. So what are they doing? They are blocking neurons, and they block conduction of information on this level from first to second cell, or on this level from second to third cell. They provide precision of vision. They block extra signals, and they provide better picture because they inhibit some neurons which correspond to borders of some objects, and it provides better precision of vision. So we have 5 types of retinal neurons. 3 main neurons, photoreceptors, bipolar cells, and multipolar neurons, and also 2 types of blocking neurons, horizontal and hemicrine cells. So we have rods and cones, and also near them we have special cells which are called pigment cells. They have processes which are located between outer segments of rods and cones, and there are melanosomes, special organelles inside the pigment cells. Pigment cells or pigment epithelium, it has basement membrane, so it's modified epithelium. It may be described as modified epithelium, and it's located near the outer segments of rods and cones. Then we have bipolar cells, multipolar cells, and all those cells are maintained by special radial glial cells. Here we can see them. They provide they maintain rods and cones, bipolar cells, and multipolar cells. They are very long, they have radial orientation, and they form outer and inner limiting membranes. Here we can see that those cells they have special feet-like projections, and they form two membranes, outer membrane and inner limiting membrane. So let's look at histological slide. We have three functional layers of retina, rods and cones, bipolar cells, and multipolar cells, but also we have morphological layers of retina, and there are 10 morphological layers of human retina. The outermost layer is pigment layer. Next, it's layer of photoreceptors or layer of rods and cones. Next, it's outer limiting membrane. Here we have this layer. Next, it's outer granular layer or outer nuclear layer. There are nuclei of rods and cones, and this layer corresponds to location of nuclei of rods and cones. And here we can see how numerous they are. It's this layer. Next, it's outer plexiform layer or outer reticular layer. It corresponds to synaptic contacts between rods and cones and bipolar cells, where nuclei are absent, but there are synaptic contacts between first and second neuron. It's outer plexiform layer or outer reticular layer. Next layer, it's inner granular or inner nuclear layer. It corresponds to nuclei of bipolar cells. Outer corresponds to rods and cones. Inner corresponds to bipolar cells. Also, there are nuclei, they have this localization. Next, it's inner plexiform layer or inner reticular layer. It corresponds to synaptic contacts between bipolar cells and multipolar cells, this layer. Granular, it's ganglionic layer of multipolar cells. We can see here their nuclei. It's this layer. Next, it's layer of nerve fibers, axons of multipolar cells, and the innermost layer is inner limiting membrane, which is formed by radial glial cells. So there are 10 morphological layers of retina. And we have 3 layers with nuclei of cells, outer and inner nuclear or granular layers, which correspond to nuclei of rods and cones and bipolar cells, and also ganglionic layer, which corresponds to nuclei of multipolar cells. Outer and inner plexiform or reticular layers, which correspond to zones of synaptic contacts between first and second neuron and second and third neuron. And generally, we have 10 morphological layers of retina. Pigment layer, photosensory layer, which corresponds to outer segments of rods and cones, outer limiting membrane, outer nuclear layer, outer plexiform or reticular layer, inner nuclear or granular layer, inner plexiform or reticular layer, ganglionic layer of nerve fibers, and inner limiting membrane. 10 morphological layers of human retina. And in here we can see those layers, and let's look at them. Number 1, it's pigment layer. Number 2, it's photosensory layer or photoreceptor layer. Number 3, it's place of localization of outer limiting membrane. Number 4, outer granular layer or outer nuclear layer. Number 5, outer plexiform layer or outer reticular layer. Number 6, it's inner granular layer or inner nuclear layer. Number 7, in its inner plexiform or inner reticular layer. Number 8, it's ganglionic layer. Number 9, it's layer of nerve fibers, and number 10, it's inner limiting membrane. And there are 10 morphological layers of human retina. And here we can see rods and cones, and let's look at their interactions with pigment epithelium. Here we can see rods and cones, and their outer segments are surrounded with projections of pigment cells. They have basement membrane, so they sometimes are described as modified epithelial cells. They have inner inclusions, which are called melanosomes. And they are movable, and they provide adjustment of eye to the brightness of light. When we have very bright light, those granules are moving and they are going to the outer segments of rods and cones. And in the darkness, they are going to the basal part of pigment cells. So this provides adjustment, and they uptake extra light to prevent damage of outer segments of rods and cones, and also reflect some light and help to accept those light by outer segments of rods and cones. So this discs and hemidiscs, those lamella, they have protection and extra light is uptaken by melanin in the melanosomes of pigment cells. And here we can see this interaction. It's pigment cell with melanosomes and outer segments of rods and cones. And another function of pigment cells that they provide phagocytosis of old and damaged lamella of rods and cones. So they provide phagocytosis of old parts of rods and cones. And here we can see rods and cones layer, layer of pigment epithelium, its basement membrane, bipolar cells, multipolar cells, and radial glial cells. And here we can compare retina in the darkness and retina in daylight. In darkness, pigment inclusions are in the basal part. Here we can see rods and cones, they are above pigment layer. And in daylight, pigment goes to the retina and it goes between outer segments of rods and cones, and it's very close to the nuclei of rods and cones, to the nuclear layer. So we can compare retina in darkness and in the daylight. And let's look at functional features of rods and cones. You know that rods, they provide black and white vision, and cones, they have more specific type of vision. They provide colorful vision. And we have 3 types of cones. We have cones which react to blue light, to red, and to green light. 3 types of cones, and they are irritated by different types of light with different length of wave. And here we can see red, green, and blue light. And you know that digital scheme of our displays in our laptops, of our phones, it's RGB type of color. And if you will take a pic from your display and will zoom those pic, you can see that each pixel of display has 3 color elements: red, green, and blue. It's RGB scheme. When all of them are turned on, we have white light. And when red only are turned on, we have red light. Red and green, we can see yellow. Only green, we can see green. Green and blue, different shades of blue, and so on. So different combinations of three basic types of light may show us all colorful diagram, all colors which we do see. And in our eyes, we also have the same type of color exception. We have rods which react, we have cones which react to blue light, we have cones which react to red light, and those which react to green light. And it provides exception of different shades of colors. So when we receive, for example, yellow light, two types of our cones are irritated, red and green. When we see something purple, red and blue cones are activated. So it's RGB, colorful type of color exception in our displays as well as in our retina. So we can see here rods and three different types of cones: red, green, and blue. Also bipolar cells, multipolar cells, and pigment cells, and glial cells. So it's our retina. And also our retina has two specific parts, two specific places. It's blind spot and macula. So what is blind spot? Blind spot is place of beginning of optic nerve. You know that there are axons of multipolar cells, and axons are going from retina. They form optic nerve, and optic nerve is bundle of axons of multipolar cells. So here we can see that they are collected, they form layer of nerve fibers, and then they excite from retina as optic nerve. Why this place where optic nerve begins is called blind spot? Because we haven't here in this place, we haven't rods, we haven't cones, we haven't photoreceptor cells, and it's place of retina where photoreceptor cells are absent. That's why retina cannot accept any information in this place, and it's called blind spot. And here we can see blind spot, it's ophthalmoscopy, and blood vessels, for example arteries, they excite via optic nerve, and optic nerve begins here, it's blind spot, and rods and cones in this place are absent. And another specific place in our eye, it's macula. It's depression in retina. It's located above blind spot, and it's place of the best vision. It's the thinnest place of retina, and also in this place only cones are present. Here we can see macula, and let's compare usual retina in other places, and in macula we have only cones which provide colorful vision. It's place of the best vision. So let's look how is it working. So when light comes here, some type of light, in this case it's green, it irritates corresponding cones which react to green light, and they are activated, and this signal grants from cones to bipolar cells, and from bipolar cells to multipolar cell, and via axon of multipolar cell this signal runs to optic nerve. It's how human retina is working. And another question, it's inverted retina. Why human retina is inverted, and what does it mean? So here we can see three basic neurons of human retina: rods and cones, bipolar cells, and multipolar cells. And usually we can think that light goes to the outer segments, and signal goes from this side to bipolar cells to multipolar cell and runs to optic nerve. But in human eye, light goes from another direction, not directly to the rods and cones. Light should come through all thickness of human retina when it achieves rods and cones. So human retina is inverted. let's look what does it mean. Human retina is inverted and light goes from another side of human retina. These rods and cones are turned to outer side of our eye. Let's look how is it working. Light comes here to inverted retina from this direction, but it passes through layer of multipolar cells, through layer of bipolar cell, and only then it comes to rods and cones. It means that a human retina is inverted, inversion of layers, and only after this rods and cones are activated, and this signal runs in the opposite direction. So signal light goes here in this direction, and after that nerve signal runs in the opposite direction through 3 neurons and goes to optic nerve. That's why we call human retina inverted. Let's look once again. Here we have pigment epithelium. Under pigment epithelium we have choroid, and then we have sclera in this part, and light goes through the vitreous body, and some light is uptaken by melanin in the pigment cells, but another amount of light goes to rods and cones, irritates them. They form nerve signal which goes to bipolar cells, to multipolar cells, and goes through axons of multipolar cells to optic nerve. And light goes here to macula and irritates it, and goes in the opposite direction. So directions of light in human retina and nerve signal are opposite. That's why we call human retina inverted. So we have 3 functional apparatus of the eyeball. We have dioptric apparatus, which provides conduction of light. It includes cornea, anterior, posterior chambers, fluid which fills them, lens, and vitreous body. It's dioptric apparatus. Also, we have accommodation apparatus, which provides accommodation of eye. It includes ciliary body, ciliary ligament, and lens. And also we can call as a part of accommodation apparatus iris and pigment epithelium, which provide adjustment to the brightness of light. And also we have receptor apparatus, which includes retina of eye. It includes receptors which react to light, and they form signal which is sent to CNS. And also there is supportive apparatus of eye, of organ of vision, and it includes conjunctive of eye, includes eyelids with eyelashes, eyebrows, lacrimal apparatus, and muscles. So conjunctive, it covers eyeball, it covers sclera, it's mucosa of eyeball, and it has epithelium which is stratified columnar usually, and some cells of conjunctive may produce mucus and fluid which covers this conjunctive. Eyelids. Outer surface of eyelid is covered with skin, inner is covered with conjunctive, and basis of eyelid is tarsal plate, which is composed of dense connective tissue, and also there is muscle which moves eyelids. It's skeletal muscle and histological structure, and also free margin of eyelids, and on the free margin of eyelid we have opening of meibomian gland. It's a modified sebaceous gland which produces something like sebum or something like wax, which covers free end of eyelid. And here we can see histological sites which show structure of eyelids. It's outer skin, but it's conjunctive of eye, it's muscle, and it's beginning roots of eyelashes, hairs of eyelashes, and tarsal plate and tarsal gland, which is located here, or meibomian gland, which produces lipid secrete like wax or sebum. And here we can see localization of meibomian glands. Here we can see them. They are located inside the eyelids. Lacrimal glands. Here we can see localization of lacrimal gland and histological structure. Lacrimal glands are like salivary glands. They contain mucocytes and serocytes, and they produce mixed type of secrete, and tears. They are produced by lacrimal glands. Then they are going to the bags of conjunctive, and then they are going to the lacrimal ducts. So we have discussed a structure of organ of vision, and in the next video we will talk about organ of hearing and also about organ of balance. That's all, and thank you for your attention.